Hi guys, my name's Ed Jarman. I work at the Institute of Genetics and Molecular Medicine in Edinburgh in Luke Bolter's lab. I'm going to talk to you today about my work looking at the role of DKK1 interactions in the tumour immune microenvironment. So a little bit of background on DKK1. DKK1 is a secreted Wnt inhibitor. It binds to LRP56 and prevents Wnt ligands from forming their normal ternary complex, which would typically drive canonical beta-catenin signaling. In most cancers, including cholangio, beta-catenin signaling is considered a tumor driver. It drives proliferation and stemness. So for all intents and purposes, you'd expect DKK1 to be a tumor suppressive protein. But in cholangiocarcinoma, this doesn't seem to be the case. DKK1 is upregulated in about 30 to 40% of cholangios. You can see in these human TMA cores, there's high expression of DKK1 in the tumor malignant epithelium. And interestingly, DKK1 in these cancers is associated with worse prognosis. So DKK1 offers this interesting paradox where despite inhibiting Wnt signaling, it seems to drive tumor progression. And this is really the paradox that we're interested in our lab. And we're trying to understand mechanistically how this works. So in our lab, we really use a hydrodynamic injection model. This is what we've been using a lot recently. The way this works is you can inject naked DNA plasmids directly into the mouse at high pressure, and these plasmids get taken up by hepatocytes. And with the inclusion of the sp13 retrotransposase, these get integrated into hepatocytes. And we use a plasmid mix which contains constitutively active Notch and constitutively active AKT. And this means that hepatocytes get transdifferentiated into a more cholangiocyte-esque cell type and oncogenically driven to proliferate. And the result is that if you don't include the retrotransposase after 4 to 6 weeks, you don't really see a phenotype. But if you include that and you get integration of these genes, you get these cytokeratin 19 positive ductular tumors multifocal across the liver, and they really resemble cholangiocarcinoma. So this is the model we're using. The reason this is a really useful model system is it's modular in nature, and you can include any genetic element or open reading frame of interest simply by adding that plasmid into the initial injection. And so for our interest in DKK1, we've included a HA-tagged DKK1 open reading frame. And this means that in these tumors, after 6 weeks, you get high levels of DKK1 expression, specifically in these tumors, when compared to the controls where DKK1 is typically low. So we've used this as a model to investigate what pathways might be altered in DKK1 overexpressing tumors that might be driving tumor progression. So we performed a nanostring gene expression analysis on DKK1 overexpressing tumors and compared this to controls. And actually, the results kind of surprised us. The main thing we saw that was regulated or upregulated was genes involved in immune modulation. So these circled in green are immune modulatory genes upregulated when DKK1 is overexpressed. And we also see in terms of a more generalized pathway score that cytokine and chemokine signaling seems to be increased. So this wasn't really what we expected, but it was a very interesting new role potentially for DKK1. And we wanted to see whether DKK1 overexpression therefore modulated immune abundance in these tumors. So we went back to tumor sections from these mice, and we looked at a number of different cell markers for immune cells in DKK1 overexpressing tumors and the controls. And one of the interesting phenotypes we saw was when we stained for F480, a pan macrophage marker, we saw a large increase in the number of these macrophages infiltrating into the tumor when DKK1 is overexpressed. You can see the DKK1 tumors on the bottom here. And this difference is quantified in the top right. This also coincided with an increase in macrophage gene signatures in the DKK1 overexpressors. So this suggests macrophages are increased in these tumors. In addition to this, we also saw a number of signs that DKK1 is promoting a tolerogenic immune tumor environment. We saw a higher number of FOXP3 positive Tregs, which are the predominant immunosuppressive cell type that regulate tolerant immune environments. And this is quantified here in the middle. And we also saw, in terms of gene expression pathways, a reduction in co-stimulatory signaling and antigen presentation when DKK1 was overexpressed. So these both point to the idea that the adaptive immune response is being stilted in these DKK1 high tumors. So we found this very interesting. And finally, we saw an increase in IL-10 expression. Interleukin-10 is the predominant anti-inflammatory cytokine. So together, a very interesting overarching immune phenotype that points to DKK1 promoting an immune quiet environment. And this might be a mechanism by which DKK1 can promote this tumor progression. So having kind of found this as a phenotype, we were quite intrigued and we wanted to look at mechanistically how DKK1 might be doing this. So we have a HA tag attached to our DKK1 protein. So we're able to pull this down via co-IP and submit samples to mass spec to see what proteins are binding. We perform this in 3 cholangiocarcinoma cell lines, human cell lines, as well as in vivo in our mouse model, in our hydrodynamic injection model. And interestingly, the one protein that we saw consistently enriched as a DKK1 binding partner in all these models was this protein GRP78, also known as BIP. So GRP78 is known as an ER stress protein. Its normal function is to facilitate the folding of newly translated peptides in the ER, and it's also one of the main factors involved in ER stress and upregulated as part of the unfolded protein response. It's not hugely novel or interesting that a protein like DKK1, which is secreted through the ER secretory pathway, would bind to a chaperone like this, but GRP78 is far more interesting than that. If you look in cancer and cancer cells in a number of different models and cancer types, GRP78 has been increasingly recognized as mislocalized and found in many different cellular compartments, but most interestingly at the cell surface and secreted from tumor cells. And there's amassing literature suggesting that the secretion of GRP78 and similar proteins have very potent immune modulatory actions in the tumor microenvironment. So I looked into this a bit further, and the reason this really interested me is the overlap between GRP78's function as an immune modulator and the DKK1 phenotype we're seeing was quite obvious. So GRP78 is known to promote, in response to ER stress, an increased expression of pro-inflammatory cytokines and chemokines. It's been shown specifically to bind to macrophages and promote cytoskeletal remodeling, leading to an increased infiltration of macrophages into the tumor, as we're seeing in our DKK1 phenotype. And also, most interestingly, from my point of view, it's been shown to interact directly with dendritic cells. And by interacting with these dendritic cells, which are professional antigen presentation cells, it impairs the ability of them to prime CD8 positive T cells, and also produces an increased number of regulatory T cells. So that indicates that it's able to stilt the adaptive immune response and promote this immune suppressive kind of cell type as well. So clearly all of these overlap quite nicely with our DKK1 phenotype. And what this means is in our research, we're now really interested in what this kind of interaction between DKK1 and GRP78 is doing in these cells. We want to answer the question whether GRP78 is responsible or involved in DKK1's immune modulatory action. And there could be a number of ways in which this is working. Secreted DKK1 from tumor cells could be binding cell surface GRP78 on myeloid cells or other cells in a non-cell autonomous manner. Or both of these proteins could be binding in the ER and being co-secreted as binded factors. Or alternatively, increased expression of an ER secretory protein like DKK1 could just be driving increased ER stress and leading to an increased secretion of GRP78 from these cells. And understanding which of these or which other mechanisms might be affecting GRP78 levels and DKK1 action in the tumor microenvironment is extremely important for how we think about potentially targeting these two factors. So our current work is looking at whether we can understand how these are interacting with the aim of potentially seeing if GRP78 can be targeted in DKK1 high cholangiocarcinomas. And we also have a DKK1 targeting monoclonal antibody, which has shown some nice efficacy. And we want to understand whether targeting DKK1 is working through disruption of these interactions and disruption of immune modulation. So these are our current ongoing questions. And with that, I'll just say a quick thank you to the lab. This is Luke's lab. Thank you to the CCF for letting me talk, and a big thank you to the Margaret Brown Fellowship for funding this research. Thank you.
Hi, my name is Meng-Ju, a research fellow from Dr. Nabil Badisi lab at MGH. It is my honor to present here and receive the fellowship from the foundation. Thank you for your support to make this work possible. Today, I'm going to talk about my project about using our developed faithful genetic engineer mouse model of IDH1 mutant intrahepatic cholangiocarcinoma to study tumor and immune microenvironment and to share some ongoing early data with you. Mutations in isocitrate dehydrogenase 1 is among the most common genetic alterations in ICC, present in 15% patients. The mutant IDH1 protein exhibits aberrant enzymatic function, reducing alpha-ketoglutarate to uncometabolite 2-hydroxyglutarate to HG. 2-HG competitively inhibit alpha-ketoglutarate-dependent histone and DNA methylating enzymes, thereby altering the chromatin regulations. The first-in-class small molecule selective IDH1 mutant inhibitor, AC20, aka ivosidenib, shows efficacy against IDH1 mutant ICC in recent phase 1 and phase 3 trials. Despite the anticipated FDA approval for AC120 for ICC, there are important challenges in attaining the full promise of IDH1 mutant targeting strategies. Not all patients receive clinical benefits. Stable disease is more common than tumor regression in those that do, and eventual progression inevitably occurs. Understanding the biological mechanism of response is critical, needed to guide improvement in treatment. Unfortunately, research in this area has been severely hampered by the absence of relevant models. So cancer-associated mutations in IDH1 virtually all involve the arginine 132 codon R132. However, the profile of specific variant differs across cancer types, with R132C representing more than 75% of IDH1 mutant ICC, whereas the most common variant in glioma R132H is found in only 1 to 2% of ICC. We generated conditional knock-in alleles of both IDH1 R132H and IDH1 R132C and crossed this strand to the liver-specific CRE strand, albumin CRE. Since IDH1 mutations coexist with activating mutations in different RAS-MAC pathway in around 30% to 40% of human ICC, we generated compound mutant mice with conditional IDH1 mutant and KRAS gene alleles. We found that R132C alleles generated much higher levels of 2-HG concentration in liver than the R132H did. So we found that IDH1 R132C strongly collaborated with KRAS to generate invasive and metastatic ICC that recapitulates the histopathogenesis of human disease, while little collaboratively was seen for R132H allele. And this finding supports the faithfulness of the murine gene model, suggests that particularly high levels of 2-HG are required to drive ICC. Next, we want to explore the response or model to IDH mutant inhibition in vivo. To this end, we use the primary tumors cultures derived from our GEM models to generate syngeneic tumor models in immune-competent mice B6, and we also inject these tumors into the immune-deficient NSG mice as well. And we found that 2-HG durably reduced intratumoral 2-HG levels by 95%. We are interested in working, knowing what the biological effect of IDH1 inhibition are. So importantly, in line with human clinical trial data, AG120 significantly slowed tumor growth, but did not yield regressions and show better survival in both subcutaneous and endoscopic settings in immune-competent mice. Interestingly, AG120 did not show effect on immune-deficient mice. It suggests that effect of AG120 appears to require intact immune system. So the result from other groups has suggested that IDH1 mutation correlates with lower leukocyte levels across all cancers, and some report also suggests that IDH1 mutation might cause the immune desert. So in our own analysis, we found that the evidence that IDH1 mutant iCCA were profoundly immunosuppressed compared with other iCCA subsets as reflected by RNA-seq of human iCCA cell lines, showing high significant enrichment of immunosuppressive TGF-beta signatures and reduction in immune-stimulatory interferon signatures. Our immunohistochemistry analysis also show reduced CD8 positive T cells in IDH1 mutant GEM model compared to IDH1 wild type GEMs. So next, we analyze the TCGA iCCA patient data by the CyberSort-X, which is an algorithm to in silico calculate the immune population in our bulk RNA-seq, and found reduced CD8 positive T cells infiltration in human IDH1 mutant iCCA. It suggests that the mutations in IDH1 may influence the anti-immunity in iCCA. Therefore, we explore the immune response in our model by analyzing the immunohistochemistry from immune-competent mice model. It showed pronounced recruitment of CD8 positive T cells compared to other immune cell population. An RNA-seq analysis also showed that interferon signal is upregulated in AG120 treated group compared to vehicle treated group. And inhibition of mutant IDH1 also increased the immune stimulatory signature and inflammatory signatures in iCCA models. So next, we use the time points that tumor volume has not changed to do immune profiling by the flow cytometry with different immune population. So it showed a pronounced recruitment of CD8 positive T cell compared to other immune cell population, and increase the level of granzyme B, which is the primary molecular mediator of apoptosis by cytotoxic T lymphocyte in tumor cells, but not in spleen, consistent with specific effect on the tumor infiltrating T cells. To prove of the concept, we also did a pilot study by directly treating the GEM model with AG120. You can see on the right-hand side, the AG120 treated mice will have more leukocyte infiltration. And then we use the immunofluorescence to examine the data, and we found that inhibition of mutant IDH1 increased CD8 positive T cell infiltration on the left-hand side and decreased the tumor-associated KICD7 on the right-hand side. So this and this is the statistics graph for the previous immunofluorescence data. So this early finding shows that AG120 acts to restore anti-tumor immunity. Here are some questions of ongoing and future work. This work will provide the foundation for strategies to utilize IDH1 mutant inhibitors in combination with other agents to achieve more durable therapeutic benefits. And finally, I would like to acknowledge my mentor, Dr. Baldisi, lab members, collaborators, and funding resources. And thank you for your listening.
Hello everyone, this is Zhang from NCL-NIH. Today I want to present my data. So the title is the gut microbiota directs hepatocytes to recruit the MDSC and promote cholangiocarcinoma. First, let's know something about the cholangiocarcinoma. As we all know, the liver cancer is one of the most common cancers in the world, and the iCCA is the most common liver cancer, and the cholangiocarcinoma is the second most common liver cancer. And the patients with cholangiocarcinoma have really poor prognosis, and the average 5-year survival rate is only 5 to 10%. The cholangiocarcinoma can divide into the extrahepatic cholangiocarcinoma, intrahepatic cholangiocarcinoma, and the bladder cancer. Next is the gut-liver axis. So this alters the interaction between the gut and the liver. There will be some secondary bioassay, nutrients, LPS, short-chain fatty acid, and the bacteria from the gut into the liver through the protein. And there will be also something such as the primary bioassay, IgG, antimicrobial molecules from the liver into gut through the biliary tract. There will be also some systemic circulation. So if there is a leaky gut, whether there will be more bacteria in the liver. So we used the biodata ligation-induced PSC mouse model. As we can find, after 2 weeks of the biodata ligation, there will be the difference of the bacteria in the gut, and they also detected the gut permeability using the feces Degrader. And even 3 days later, they could find the increased gut permeability. And in the liver tissues, there will be the increase of the myeloid cells. Next model we used is the DSS-induced colitis. So we treat the mice using the DSS in the drinking water. After the treatment, you can find the colon lungs decreased and the normal epithelial cells can be destroyed. And in the liver tissues, you also could find the bacteria. So firstly, we use the biodata ligation-induced PSC mouse model. We first detected the gut permeability using the feces Degrader. So we could find the increased gut permeability in the BDL PSC mouse model. Next, we cultured the liver tissues, and we could find the increased translocated bacteria into the liver tissues. And also, we detected the 16S iron levels in the port vein blood. There will be the increased 16S iron levels in the BDL PSC mouse model. Next model we used is the DSS-induced colitis mouse model. We do the same thing. We found the increased gut permeability, increased bacterial translocation into the liver tissues, and the increased 16S iron levels into the port vein blood. We also detected the bacteria using the fish. We used the UB338 as a probe, and we found the increased bacteria after the DSS treatment in the liver tissues. Next is the cholangiocarcinoma mouse model. We using the hydrodynamic injection of the oncogene plasmids Notch1 and AKT. After the injection, about 3 to 5 weeks, you could find the cholangiocarcinoma. Another model is also the hydrodynamic injection. We use in the YABA and AKT. About 5 weeks, you could find the tumor. So we first combine the AKT-YABA cholangiocarcinoma and the biodata ligation. We could find that biodata ligation can promote the cholangiocarcinoma, and the DSS-induced colitis can promote the AKT-YABA cholangiocarcinoma. DSS-induced colitis can promote AKT Notch1 cholangiocarcinoma. Next, we want to know why. We firstly, in the biodata ligation induced PSC mouse models, we found that there increased MDSC in the liver tissues, and the ability of the MDSC to inhibit CD8 T cell proliferation is also increased in the after biodata ligation. Next, we want to know if we decrease the bacteria in the gut, whether the MDSC will decrease in the liver tissues. So we use the neomycin to decrease gram-negative bacteria. We could find neomycin treatment can decrease the MDSC accumulation in the liver tissues of the BDL-PSC mouse model. Also, neomycin treatment can decrease the MDSC accumulation in the DSS-induced colitis mouse model. Next, we want to know if we give back the bacteria, whether the MDSC will increase. So we use the vancomycin or neomycin to kill gram-positive bacteria or gram-negative bacteria. And then we collect the stools and give the stools into the germ-free mice, and then we detect the MDSC. Because vancomycin kills the gram-positive bacteria, so vancomycin stool, they have the gram-negative bacteria. Vancomycin stool treatment can increase the MDSC in the germ-free mice liver tissues. So this is we can see the gram-negative bacteria lead to MDSC accumulation in the liver. Because we know the IPS is the product of the gram-negative bacteria, and the TR4 is the receptor of the IPS. Next, we want to know the effect of the IPS on MDSC accumulation. So in the white-top mice, IPS treatment can increase the MDSC in the liver tissues, but it doesn't work in the TR4 of the mice. And DSS also increases MDSC, but it doesn't work in the TR4 of the mice. Next, in the tumor Marion mouse model, neomycin treatment can decrease the BDL promoted AKT-YABA cholangiocarcinoma, and neomycin treatment can decrease the DSS promoted AKT Notch1 cholangiocarcinoma. As we all know, the DSS can promote the cholangiocarcinoma in the white-top mice, but it doesn't work in the TR4 of mice. That means the cholangiocarcinoma development is TR4 dependent. And next, we want to know where the TR4 is from. So we use the conditional knockout mice. We deplete the TR4 on the hepatocytes using the albumin Cree. After the TR4 depletion on the hepatocytes, we could find the cholangiocarcinoma decrease. So that means the TR4 on hepatocytes mediate the cholangiocarcinoma development. In summary, we see if there is the leaky gut because of the liver disease or colitis, there will be the microbiota or the IPS go into the liver through the port vein, and they can stimulate the hepatocytes through the TR4, and this can recruit MDSC, and finally the MDSC can promote the cholangiocarcinoma development.
All right, so today my topic is development of the FGFR degrader. I'll give you a very brief introduction. My background, I come from Dana-Farber Cancer Institute. I was trained as a medicinal chemist. give you a brief introduction about this target, FGFR, called fibroblast growth factor receptor kinase. So FGFR is one of the targeting receptor kinase which can get activate many different signaling pathways that including the PR3K, AKT, and also the MEK pathway for the ERK. And the cell seems really relate on this signaling pathway, especially for the proliferation, survival, and also differentiations. Overexpression and the application of this, amplification of this target has been linked to many diseases, that including cancers. For example, breast cancer has about 80%, 20% about the FGFR amplifications. For the liver cancer, even 45% of the FGFR2 fusion proteins, and also many other cancers. That's why the target has been very attractive for the medicinal chemist and for drug discovery. So far, this concept of the small molecule has been discovered and reported either as a proof as the FGFR inhibitor, the investigational proof. Some of that has been clinical trial. And last year, the G compound, which shows on the top left, has been approved as drug for the cancers. But however, all of these small molecules don't show any good activity between these FGFR isoforms, because FGFR have the 1, 2, 3, 4 isoforms. Each of these isoforms may have the different of the functions. So to get the selectivity among these isoforms has been regarded as very important for the therapies. However, because of this FGFR isoform share very high conserved ATP binding pocket, so it's very difficult to get the selectivity between these isoforms. So far, there's no selective inhibitor between among these isoforms has been identified. So recently, there's a new technology called PROTAC, which can induce a protein degradation. The principle for this technology is you have the ligand for the target protein. For example, you have the binder for the FGFR proteins. And also you have the binder for the E3 ligase proteins. Then you put the linker between the two molecules and make a conjugate of these two molecules. Then you can recruit the E3 ligase to your protein that you want to target. And then this E3 ligase can recruit another protein called E2 ligase, which can further ubiquitinate your target the protein. And then once your protein gets ubiquitinated, then the protein will go to the proteasome machinery. Then your protein will be degraded. So that's the theory for this technology. There's quite a lot of small molecule has been used for this PROTAC technology to degrade kinase protein. For example, in our lab has been used this technology for the CDK9 proteins. One of the beauty for this technology is actually the compared to the inhibitors, this technology can really have the high selectivity even for the same protein target, but within the isoforms. This has been found that with the PROTAC technology, you might have this selectivity between these isoforms. So for E3 ligase ligands, currently there's not many. Just a couple of the E3 ligase has been discovered with some of the ligands. For example, this slide just shows some of the examples, which include VHL ligand, Febron ligand, which has 3 compounds called thalidomide, pomalidomide, and lenalidomide. And also IAP ligand shows on the right panel. There are 2 molecules shown here. These just some representative molecules. There's also some other ligand for other ligases, for example, MDM2 ligase. But overall, all this ligand shown here has been frequently used for the PROTAC molecule. That's why I listed this molecule here. So the rationale for design of the FGFR PROTAC is shown on the slide 6. So we have the FGFR inhibitor I showed in previous slide, also shown here in the left panel. This also just representative of the inhibitor for FGFR proteins. And in the middle, you can see there I show 2 examples of the linker. You can use the either alkyl linker or the carbon linker here, or you can use the PEG linker with oxygen insertion in the middle. So you can use a different length, different composition of the linker, and then you can link 2 molecules together with the left FGFR inhibitor to the right part, right hand of the ligase ligand. Show the right panel here. Then you can make the PROTAC molecule showing on the bottom. We call the PROTAC series 1, 2, 3, 4, which the compound I'm going to show in the following the slides. So with that strategy, we make the series compound. The first series compound I show here to check if whether this compound can induce the FGF protein degradation. We choose the BAF3 cells first, which have the TL FGFR2 fusion protein. So we do the wet and blot to see if the FGFR protein can be downregulated. You can see here upon the compound treatment, 2 compounds called 37 and 38. You can see with the 1 micromolar FGFR2 protein, actually this is the fusion protein can be reduced by the 2 molecules. However, once we transfer this compound to the cattle cell, which is the cancer cells, you can see that gastric cancer cells, you can see that FGFR seems to not get reduced. So it's really confusing at that time. We don't know why the compound can degrade the target on the BAF3 cells, but cannot degrade the protein on the cancer cells. Then we make the second series compound. Again, it happened the same as the series 1. You can see the 2 compounds, compound 73 and 74, also show the degradation with the 1 micromolar concentration at 24 hours. However, we didn't see that same effect on the K23 cells with 24 hours time point. Yeah, it was confusing at that time, but we still don't know. Even now, we still don't know why this compound behaved differently in 2 different cells. Then we make the series 3 show here. We only made 3 compounds show here. This series compound don't degrade the FGFR protein in the BAF3 cells as well as in the K23 cells. So basically, they are all negative compounds for the degradation of FGFR proteins. Eventually, we found 1 series compound called series 4 compound. We found that the compound indeed can induce the degradation of the FGFR2 protein in K23 cells. You can see here with 1 micromolar, the compound called DGY-9-192 compound, as well as NGH compound called the 5-79 compound, induce the obvious degradation of the FGFR2 proteins. So we're wondering what's the mechanism for this degradation. So if we pre-treat off the inhibitor, the cells, then you treat the degrader. You can see here you pre-treat the FGFR inhibitor with 10 micromolar. You're going to block the degradation of the compound called DGY-9-192 compound. Also, if you use the proteasome inhibitor called bortezomib, with even 500 nanomolar, you can see that you lose the degradation of this FGFR2 protein. Also with MLN4924 compound, which is a methylation inhibitors, also you can see that you can rescue this degradation of the FGFR2 protein. This tells us that this degradation is dependent on the proteasome pathways. And also we look at the dose dependency. You can see that even with the 50 nanomolar, we start to see the degradation of FGFR2 protein. And with 500 nanomolar, you basically completely degrade this protein. And finally, we also look at the time course. With the shortest time with 4 hours, we start to see the degradation with 100 nanomolar. And also with 8 hours, you see the same effect. 12 hours, still see the degradation. However, with long time, it seems low efficiency with 100 nanomolar, probably because of the recent size of the FGFR protein. But overall, you can see that the compound can induce degradation very quickly with short time points. Then we make the negative control compound, which is the NGH5 day 21 compound. This compound cannot binding with the E3 ligase, so we put an additional group to this E3 ligase ligand, which blocks this E3 ligase binding. So this compound only serves as the inhibitor, not the degrader of the FGFR proteins. You can see here in the K2-3 cells at 4-hour time point, only DGY compound induces degradations. However, this negative control compound doesn't induce any degradation. Again, this indicates that the protein degradation of the FGFR2 protein is actually the E3 ligase dependent. And also we test the compound in the cell growth inhibition. So you can see compared to the negative control compound, our DGY, the PROTAC molecule, shows very potent cell growth inhibition with 1 nanomolar IC50s. However, the negative control compounds have about 70-fold shift of the potencies. Again, they tell us that this cell killing effect is actually mostly comes from the degradation rather than the inhibition of the FGFR protein. To look at the selectivity of the degradation among these FGFR family, we use the CCLP1 cells, which has high expression of the FGFR1 proteins. You can see here, we saw that this dose-dependent degradation on the FGFR1 protein as well. We also use another control compound, which is a known CDK9 degrader, just use as a control compound. See that at the same conditions that we can see the degradation of CDK9. On the right panel, we see that we also test the FGFR3 and the FGFR4 proteins in the GHH7 cells. You can see that with the same dose, we didn't see that degradation for FGFR3 and the FGFR4. They tell us that this is selective. The compound have the selectivity for the FGFR1 and 2, but against the FGFR3, FGFR4 in terms of the degradation efficiency. And also we look at the FGFR1 protein degradation with the time course. You can see very similar to the FGFR2 protein. FGFR1 can be degraded by this compound at a very short time point, even 2 hours. And then next, we want to see how selective of this compound in kinase gains basically tell us, beyond the FGFR proteins, any other kinase actually being inhibited. It not necessarily can be degraded, but it can still be inhibited, especially this in vitro. This panel includes about more than 400 kinase. This is just binding profile. You can see that compound should quite well of the selectivity. The top target is one of the EGFR and FGFR proteins, and not many other off targets. Finally, we take one of the compound into the in vivo study, the PK study, to see how stable this compound in terms of the metabolites in the mice. You can see that we do the IV dose and IP dose. compound showed quite well the stability with half-life from the 4 hours to 5 hours, either IP or IV route. And with this, and we see quite good of the AUC level as well. So I think the compound should be stable in the mice, and then that's going to help to design the in vivo study for the efficacy study. This will be the next plan. So for the future, we have to make the more potent compounds, even though we saw that the degradation has been very efficient, but we think we probably can further improve the degradation efficiencies. Especially, we need to make the more selective FGFR2 degrader to get rid of the FGFR1 target. The reason for that is quite a few papers indicate that FGFR1 protein is the inhibition is probably the dose limited toxicity has been found for the many known pan-FGFR inhibitors. So the next goal is to get the selective FGFR2 degrader, but not the FGFR1 degrader.